rehabilitation centers to enhance bat care, research, and conservation.
Awesome. Well, thank you all. And thanks for putting up with the Zoom here. I'm over in Snell and I don't think I'd make it over there in time. So yeah, well, good afternoon, everyone. So I'm Rachel Handy. I'm one of the academic advisors here in IB, and I'm excited to share with you about the potential opportunities that wildlife rehab centers present towards the biology of bats and their conservation. Just an FYI, I completed my research. This was part of my master's thesis while I was still in Ohio. So a lot of the stats will be kind of Ohio oriented, but I did throw in some Oregon stuff here too, now that I'm out here. But yeah, let's dive in. So, all right, if I can move this. Oh. There we go. Okay, so I'm sure that some of you are aware that many species of bats are in trouble for a variety of reasons, from white nose syndrome to wind turbines. But what many of you might not realize is that there's a really unique opportunity to learn more about these challenges by teaming up with perhaps a surprising partner, the Wildlife Rehab Center. So I'll give you some kind of background about my research. What we know is that bats are facing a wide variety of threats, yet they are still poorly understood. Bats also are frequent patients at wildlife rehab centers, and these centers offer many opportunities to advance knowledge of bat biology via collaboration. Unfortunately, there are many negative attitudes and behaviors towards bats that have been cultivated over time. Just a few of the many myths and preconceived notions about them include, getting caught in your hair, being blind as a bat. They're often associated with evil or diseases, thought to be flying rodents, even though they're not, in fact, closely related at all. But what we do know to be fact is that bats have an immense impact on our lives, and many of us don't even know it. They have tremendous economic value, estimated in the billions of dollars. They're social and intelligent, and they have intrinsic value. They're just neat and unique creatures. Fortunately, over time, many groups have created educational outreach campaigns, such as, Bat Conservation International, about bats and their positive qualities. And this has led to an upswing of engaged citizens. I was fortunate enough to experience that on a daily basis during my time working at that rehab center in Ohio. However, new challenges are still emerging, every day. So what are some of these things that are happening to our bats? Sadly, across the globe, they are in decline, both from natural and anthropogenic causes. A few of these threats that bats are facing include habitat loss, wind energy development, diseases like white nose syndrome, colony disturbance, and invasive species like non-native plants and feral cats. And that's kind of just the tip of the iceberg. So this is where rehab centers come in. Rehab centers are located across the globe, including all 50 states here in the US. But what is it exactly? So rehab is typically considered to be broken down into 3 phases: rescue, which can be targeted or opportunistic, rehab, and then release. And the goal really is to provide that temporary care for injured, sick, or orphaned wildlife with the goal of releasing them back into the wild. And that's per the International Wildlife Rehabilitation Council. So how do our bats fit in? So, in some cases, the numbers of bats that are entering these centers is quite staggering. So for example, at the Ohio Wildlife Center where I worked, over a 15-year period, a total of 777 bats, over 7 different species, were admitted, of which 33% were rehabbed and released. Additionally, over the years, the number of bats entering these facilities has risen in many cases. So for example, in 2019, they admitted 220 bats, which was a whopping 95% increase from the previous year. And then in 2020, about 140 bats were admitted, of which 35% were released. For comparison, in 2019, at one wildlife center here in Salem, they admitted 30 bats, of which 10% were released. So with the number of bats entering these centers climbing, these facilities are well positioned to contribute to bat conservation, especially since proactive approaches to conservation are far more effective than reactive strategies. So some examples of research opportunities include data collection, more on that in a second, physical bats in hand, of course, that we're working with, and biological materials. So there's a plethora of these biological materials like guano or fur clippings that these rehab centers are pretty much throwing away on a daily basis. So what about this data collection? Wildlife databases are a really useful tool because almost all rehab centers are required to keep track of patient and presenter records to maintain their permitting. So one example database I'd like to highlight is Wild One. This is a free system created by the Wildlife Center of Virginia. And this database currently has at least 107 active participants across 5 countries. That's a lot of information being collected, which has the potential to reveal emerging threats. However, this data has not been well mined yet. So for those wishing to collaborate with rehab centers, it offers researchers a chance to gather information and really hit the ground running. So a couple of stats I want to highlight. Again, in 2019, a total of 2,797 total bats were entered into these databases, 40% of which were released. This included 16 different species across 26 organizations here in the US and Canada. So that brings me to collaboration and partnerships. What can we do with this information? So collaboration between these centers and potential partners could really enhance our knowledge of bat biology. Furthermore, because many of these conservation concerns threatening bats are common across many species, collaborations on studies of bats can really have far-reaching effects. Example collaborators might include scientific researchers, so the data points I mentioned earlier. This one hopefully is ringing true to all of us here, our educational partners working with local university students, either undergrads or graduate students, and of course our veterinary and vet tech students too. What about governing agencies and policymakers? So as I mentioned, rehab centers need to be permitted, and those agencies that are providing the permits are a great partnership to work with. A lot of times they do provide various protections for different endangered or threatened species, and they have a huge impact on public perception as well. Additionally, the community is a huge resource and a great potential partner. As I mentioned, I had literally thousands of interactions on a daily basis with people when I worked at the rehab center, and all those one-on-one interactions allowed me to educate them on that basis. In a more formal way, rehab centers can provide different educational programs to schools or members of the community. And of course, the community provides us with activism and volunteers too, which are critical. So some potential research questions. There's numerous, probably an infinite amount of things we could think of, but a few I want to highlight or that I've thought of include: what are the most common injuries and sources of mortality for bats entering these centers, and how do they vary across landscapes? What about pesticides or other chemical toxins? Which ones are present in bats, and do they vary by foraging strategy or location? And one I was particularly interested in: what is the post-release survival rate of rehabbed bats over the short and long term? And do those rates vary by species, injury type, and geography? All right, and with that, I'd like to thank you all for attending. If you are interested in learning more, I'd be happy to provide you with a link to my full presentation. And then my findings were also recently published in the Journal of Wildlife Rehabilitation this past January, so I could provide that as well. But thanks all for listening.
Rachel, anybody have questions? I have one, Rachel. We've been interested in using data from, where's the camera there? Oh, there, okay. From wildlife rehab places for birds and heat waves. And one of the challenges is that they collect data in really different ways, or they don't collect data that would be like critically important for a research question.
Yes.
Would you like work on that kind of a problem, and you have tips?
Yes, absolutely. So that was a huge part of my findings and my experience too, working in these centers. So, you know, especially because wildlife centers are so heavily reliant on volunteers, it's like everyone does everything a little differently. It's kind of hard to enforce, you know, the way that our scientific minds work of like, we need to have everything, you know, unbiased and equal, and we need to collect data in a very specific way. And it doesn't always work out that way. So in my paper, I did provide some suggestions on how both the scientific community and wildlife rehab centers can work with each other and be aware of different things to consider in order to kind of help with some of those issues. So yeah, I encourage you to check out my paper and you can learn more.
That's good. Thanks, Rachel.
Yeah.
Other questions? Yep, yeah, one.
Yeah, this is Dee. That was awesome.
Hi, Dee.
That seems like a really large number of bats getting into these facilities. I'm curious, do you know like the circumstances? Is it kind of bats getting into people's houses and things not going well, or
Yes.
reasons why so many bats end up in facilities?
Yeah, that's a great question. So where I was in Ohio, we have all insectivorous bats species in our state. So those bats are the ones that are typically, you know, hibernating over the winter during the colder weather, and then, you know, emerging for the spring and summer, fall months. So for us, a large majority that came in, as you mentioned, would be that they're showing up in people's homes, kind of in places where they're not supposed to be, with these wildly changing weather factors that we have. So it's staying warmer longer. Maybe we'll have a cold snap and then it gets warm and they wake up. They're just having all these issues and they're showing up in places where they're not always supposed to be. So that was definitely a common thing for us. Other issues, as I mentioned, you know, outdoor and feral cats are a huge problem. So we have a lot of pet interactions a lot of times. That was definitely common. And in one of the recent years, again, with kind of the climate and weather impacts, we had major flooding, and it caused some issues with an uptick of orphaned bats because of the way that they were getting, you know, separated from their moms with these floods. Their hibernacular were being affected. So we had like a huge upswing one year because of flooding issues, but it's a pretty wide variety of things. But for us in Ohio, those were kind of the main factors. And then white nose syndrome was huge as well. So that was also an issue. But again, typically with that, it's, you know, they're waking up at times that they shouldn't be and being found and then being brought in.
Awesome. Thank you so much, Rachel.
Yeah, thank you.
Really appreciate you sharing your work with us. Super cool.
Happy to do it.
Okay, so we're going to move to Nate Kirk, who is going to tell us about a cure to aggression. Leveraging undergraduate research classes to understand competitive interactions in the Rocky Intertidal. Thank you, Major.
So yes, you always have to have a joke title. And I convinced one of my students to do this, student I'll highlight later, and they didn't want to do it, but they ended up getting a job offer at their poster, and part of what they liked was them. Anyway, so yeah, I want to talk to you a little bit about what I've been doing out on the coast with sea anemones and aggression, and C461, which is an invertebrate zoology class based out at Hatfield. And so cures are kind of a topical thing. We're talking about it as a faculty, as a way of being able to bring our research into classrooms and have students be part of it. What makes it, I like this framework by Cissy Vallon at Auburn, where it's relevant, where at the end, I actually care what the data that the students are doing because it's relevant to my own research. I can't advance. All right, that's no worries. Okay, thank you. Yeah. So there's all sorts of benefits of these, increased skill sets, career aspirations, things like that, retention in science, graduation rates, and basically what it comes down to is I could probably effectively mentor 2 to 3 undergraduates working in my research at a time, or I can somehow try to get 30 people involved in these larger scale projects. And so as proof of concept last year, a really talented postdoc, Sam Budgood, helped teach this class with me, and he had one additional thing. He also works on Anthopleura elegantissima, one additional figure that he needed data for, and he could go out and collect it. But instead what we did was we had our students do it. So we went out there, we collected surface area, volume ratios, looked at chlorophyll, and basically this is them the first day of class, really getting in there and collecting our animals and doing this. So basically the moral of the story is he's going to submit it very shortly with a consortia author of all these students that were involved, and they're going to get a publication out of it, and he had less work to do to get his own publication out. So win-win. So initially, I was annoyed that we were denied permitting a lot of places except Seal Rock, which is a major recreational place, a state park. But now I've actually grown to love it, because if you bring power tools to the intertidal, people come up and want to ask you about what you're doing. And so typically my students do my work while I run interference, and I get to talk to lots of kids and people about what we're doing out here. But this is Seal Rock. At the far end is our rock. And on the rock, what we've been doing is we've been marking these colonies. And here, what we can see is I've outlined them. Here are our particular colonies. And we can see that there are spaces between them. And we know what causes those spaces between them. They're actually incredibly aggressive to each other intra-specifically. So they will fight each other. I'll show you some examples and some videos in a second. But basically, what we're interested in is these are aggregates of animals out here. They're actively fighting, which causes these lines of demarcation between them. But what's the end goal, right? What's this rock gonna look like in 5, 10, 20 years, or whenever I get fired, right? So the longer I stay here, the more interesting this talk will become, just in case you're thinking of firing me. And so sex and aggression and all of these things are things that the students are really interested in data on, and then lead to our sort of molecular stuff. So there's a couple of things to talk about in terms of the basic biology to help make this sense. One is that they can divide and conquer. So they can make clone armies of themselves by literally ripping themselves in half, which is actually experimentally kind of aggravating because they tear apart their mouth in the process and can't feed. So Sam wanted some feeding assay data, and all of them started to divide, and that was a problem. But you can see what happens. They just literally rip themselves. That's the same animal. And then they pull themselves far apart, and then they rip. And then they have to heal and repair themselves, and now you got 2. And they do this all over. But they can also have sex, because they're either male, they create male gametes, and that we could tell at a light microscope, dissecting scope and a light scope, or they make eggs. So sperm or eggs, never sperm and eggs. Many cnidarians can do that, but we've never seen that. And in a colony, it seems to either be all creating sperm or all creating eggs, not mixes in a single colony. So we think it might be genetics that controls this, but we don't know. And we're also in the literature, there's also a really hard stop in the late 80s where there was a lot of stuff done on the ecology, and then not much after that. And you can go into the literature and we see very skewed sex ratios, not the 50-50 male-female that we might expect. We're seeing like 80%, 20%, 30%, so on and so forth. So skewed ratios. Some of them are small sample size issues. I agree, but this was something that really interested a student, and they followed up with this. And so here is our field site down here. We've got a number of different colonies tagged here throughout this. And if we go to our rock, we have egg or sperm or unknown. Some colonies, they have to reach a certain size before they'll create eggs and sperm. Some divide before that. So you end up with all these colonies which do not make eggs or sperm. So they are, for whatever reason, that year, the first year we went out, not undergoing sexual reproduction trade-off. So here is our rock here. And if we go out into our rock, we've been tagging colonies. That is colony 39. I could tell you, thanks to Jean, and I'm totally grateful to Jean for this. They wanted to do this project. There are 973 anemones in this colony. It takes, that's the tidal height. It takes up about 2 meters of this rock. And we have this for all the colonies on this particular rock. Right now, we're doing fighting matrices. I'll talk about that so we can get an idea of their competitive ability and their aggressiveness so we can start to make predictions of what the rock should look like in our opinions. So this is Sam out there putting down Z-spar. It lasted about a week out there. So this year I went back with my son Norman, and we went out and drilled into the rocks. We'll be able to permanently follow these colonies. Some of these colonies looked exactly like what their pictures were from years past. Some of them did not. Okay, so there are changing even at a 1 year range. So a number of interesting things popped out. We have sort of an even sex ratio, but we can see that they're sort of grouped. These are all males, these are all females, and here is a group of females. And what makes the most sense is that, sure, they just like a couple clones wandered away and formed another colony. But as we interact them together, they're fighting. So either they've lost the ability to recognize that they're clones or they're not clones. Okay, so we'll fill out that, but right now that's still a puzzle. And it seems to be highly significant that they're clumping together. Also, we have how many there were last year. We just went out. I don't have time to talk about that, unfortunately. And we can see that there are differences, but we can find our colonies again. So we'll be able to follow these over time, especially now that we've tagged them. And you might notice that that's gone now. So some of the colonies are changing shapes. We don't know if they're getting bigger or smaller, but there seems to be some winning losers. We can bring them back to the lab. We can collect them. We can put them in here. We can settle them out on tiles so we can manipulate them and fight them. And this is great. I got an email. The title was A.L. Antisima as a bully. So this is Xanthogrammica, a congenitor. They were size matched, which means that this is infant and this is an adult. But what we can see is these giant acaragi full of nematocysts that are going to whack each other. And this is what they do. They sweep them up and they bash each other. And if you look left behind some of that tissue. Isn't that awesome? An undergraduate at Western followed up on this. And so that is all nematocysts that will continue to sting after the fact. And they will kill each other if you put them together for about 6 days. I didn't do those experiments, but it is out there in the literature. And so there's a couple of things, right? One competitor could kind of duck after a certain amount of time, and we could figure out like how long that took. Or one might just start to wander away. So I co-developed with an undergraduate an ethogram that we've been able to then use. And we've even had eCampus students scoring videos as part of research experiences, which I'm hoping to move into a different course. Elise was a student in the class. She decided to go above and beyond, and she was really interested in like the repetitive fighting, like what happens if you put them every day. So this is just the number of tentacle hits that occurred over an hour. We see fight 1, not very much. Fight 2 got more. Fight 3 got, and then we gave them a break, and it dropped back down. Okay, so if you fight them, they get more and more fighty, and then if you give them a break, they return here. The same thing for the acaragi hits, but that was not significant. I think it's a sample size issue. But this was really fascinating data. So these are the fights down here. This is the number of the time until the first one ran away. Okay, and you can see that it seems to be getting less. But what's really interesting is in the first fight, every single one ran away eventually. In the second fight, 5 out of 12, 2 out of 6, and the last one, they held their ground. Yeah, that one ran away really fast, but almost everyone else stood for the entire hour and fought each other, which is really kind of cool stuff. So we're going to follow that up. In terms of them getting ready, they started off with no acaragi, those fighting tentacles, and then over time they got more and more and more ready to fight. So the ones on the outside are definitely ready to go. We also found a slight effect of colony, but we have way too small to actually see if it's real. That's what we're going to follow up with this year. Going forward, there are incredibly few downsides to involving your undergraduates in your work. Everybody benefits. We have 30 people out there helping us collect these data. And as a molecular ecologist, we've got genetic samples for rad. I just found out that in my CQLS directories, we have a genome of this thing that just needs to be annotated. Holland Elder made it a couple of years ago. So we'll be able to then do that and then start to pair some of the genetics with the ones like we know this clone's really good at fighting. What makes it really good at fighting? So that's sort of where we're heading. This is the fastest I've ever talked,
Yeah.
but I made it.
Awesome. That's really cool, Nate. Question?
Thank you. I agree. Yeah.
I think I have an annotation for Anthura that I did for pollen. You should never use this.
Nate, it just keeps getting better.
All this stuff existed. So part of the reason why I'm so thrilled about talking to you today is I would love collaborators. So if you're interested in any part of this work, let's chat and we'll get 30 people to do our research in a couple of months. Yeah.
So when you were doing those, the lab challenges or whoever did those,
Yeah.
were those, did you like pit the same individuals against each other multiple times?
That's it for the repetitive.
Yeah.
That is exactly what we did.
Okay, so they
So
knew each other.
They knew each other. Yeah, totally. We would move them apart, we put them back together, we move, and so
Yeah.
they got to be friends. And then at the end, we put them
Frenemies,
together,
call 'em.
frenemies, yeah. Well, they didn't run away from each other, so
Yeah.
they were like sparring partners. But we put them together, and this was a miscommunication. We meant to do a self fight first. And
Clone on clone.
clone on clone to make sure that they don't fight each other. And so we've that did not happen. And then at the end, I was like, well, just grab a clone from that and put it together. And most of the time they just interweave their tentacles and it was no problem. But near the end of one of these fights, all of a sudden, one of them just blew up their acroagi and just got ready to strike, but then didn't. So I'm wondering if little bits of macroagi and something else
Were
got left
detected,
behind
yeah.
and it was sensed it, touched it, but then when it reached out, there was
Yeah.
no connect. And these things have no centralized nervous system, so it's all reaction. And so as a behavior, looking at the behavior of it, you get rid of all the thinking. And that's amazing to be able to distill it down to just these simple behaviors based on just their genetics and other cool stuff.
Yeah, so are you guessing there's chemical
Yes.
indicators of
Yes.
self or
And there's
whatever, yeah.
something else that started in the 70s and then was abandoned was this thing called anthoplurin. It has some awful name, a chemical name, but basically it is I forget what the chemical that goes from one individual to another one, pheromone?
Yeah.
Pheromone. So a pheromone goes out there, and then they start to react. When something is hurting an enemy, you can drop it in the water and they start to do that. But we don't know if it's the same clone. If you take it from a closely related clone, is it have a stronger reaction? So that's all. I want to follow up on that because
Yeah.
undergraduates can easily do that. It's a simple grind it up, get the stuff and drop it and see.
Yeah,
So there's all sorts of cool stuff.
Yeah, I kind of relate to that.
Yeah.
I think thinking of some of these colonies are very large.
Yeah,
And I'm assuming as they divide, they stay put. So the distance is associated with how long ago they started, the farther away they are from each other, maybe there's some isolation by distance.
Yeah, so Sam is actually really interested in that. So he how old is this colony, right? Because there's in the literature, it's these things are 100 years old. But if so, these are clones. And so you can look at mutation rates and start to see
Well,
do they not?
Recognition,
Yeah.
might break down. as
Yeah.
people away from each other.
Yeah, that's cool.
Because in Hydra, that's totally true. It's these multivariable loci that are controlling whether or not they recognize each other as self and non-self. So absolutely, that could be in play.
but they do have some mobility, right?
Oh, yeah.
Yeah.
Yeah. So they'll move
moving.
around and they put them into this division of labor. There's a number of studies on that. And then individuals would wander off, they call them scouts, and they would get beat up and they would come back and they get beat up and they go.
Hard
And
life,
and it's and this because they have no centralized nervous system that they'll get beat up and then they'll just circle back around and get beat up again. They're just kind of wandering aimlessly. But yeah, some of them, and I think that does create population. So I think we do have clones, and that's why I need to do all the pairwise fighting to see which ones are likely clones, and the genetics will also shake that out. But yeah, those are next steps. But yeah,
Awesome,
yeah,
thank you, Nate,
Yeah,
that's
thanks.
really, really cool stuff.
yeah, I agree. I like this.
Also, the math seminar that Mark Novak sent around recently had really cool modeling of similar stuff, but in wolves, how wolf packs have boundaries, marking, to get territorial boundaries. And can you put all kinds of different factors into those models? So it might be at some point. Sounds like a cool place to go with it. Okay, well, next we have Sylvia, who's going to tell us about European green crabs and whether or not they are here to stay. And she brought show and tell.
Thank you, Jamie. It's a pleasure to be here. How many of you have seen green crabs on the Oregon coast? Very good, very good. Do you know how to tell? Let's see. Oh, okay. The best way to tell green crabs from our native crabs is not by color, but by the spines behind their eyes, 5 spines. None of our native crabs have 5 spines. For example, the Dungeness has 10 small ones. Now, the success of the European green crab on our shore, be it range expansion or abundance in our estuaries, is linked to ocean conditions of their experienced by their larvae. And what are these ocean conditions? Well, first of all, the larvae will not develop below 10 degrees Celsius. So also they need favorable currents to take them to suitable habitats. And the favorable current on our coast for green crabs happens to be the Davidson current. Now, the Davidson current is a very strong current. It originates in Southern California in the winter time, and it hugs the shore, moving northward all the way to Southeast Alaska. You can think of the Davidson current as a giant conveyor belt moving warm water masses northward. And in those water masses, there are plankton, right? And also southern copepods and larvae. They can get traveled quite far. So here we have the Davidson current is especially strong and warm during El Nino events. For example, 12 degrees Celsius in the winter off our coast is not unusual. Also, it can last a long time during El Nino events, from September to April the following year, a total of 8 months when a larvae could be transported northward. That's pretty impressive. And also, average velocities can be 30 miles per day. And if you're a larvae in the water mass, and it takes maybe 60 days for you to complete your development, you can be transported over 1,000 miles. Now, I should mention that we had 2 very strong El Niños in the recent past. The 97-98 El Nino, green crab larvae made it to Oregon, Washington, and British Columbia and colonized those places. And then 2015 and 16, they got into the Salish Sea, the inland sea between Vancouver Island and the mainland. Now, this is a representation of their range expansion. Green crabs were first introduced into San Francisco Bay during probably the 1980s. They were introduced from the East Coast along with seafood products, maybe lobsters or bait worms that were wrapped in algae. And there were a lot of hitchhikers on the algae that actually got established in San Francisco Bay that way, including the green crab. And from San Francisco Bay is a huge bay with lots of sloughs where there's not a lot of water movement. And the green crab larvae were retained, the population built up. And any larvae that made it to the open coast would just hitch a ride on the Davidson current and get transported northward. Okay, this is a representation of the range expansion along our coast. And what I want you to note is that it was not gradual. There are periods where nothing happens, and then there are periods of episodic range expansions. And those happened mainly during El Nino events. Here we go. Here we start in San Francisco Bay, getting to Northern California, Southern Oregon, and now we have an El Nino, and they make it to Vancouver Island. Not much is happening now. They make it to the central BC coast, and now we have an El Nino, and they make it into the Salish Sea. Now they're in Haida Gwaii, and last year they were in Southeast Alaska. So it didn't take them very long, just a few decades to make that range expansion. Now let's look at the abundance of green crabs in our estuary. Like after the 97-98 El Nino, during the winter, there was a great opportunity for larvae to be moved northward. And all these estuaries that we checked, we had young green crabs And at the end of their first growing season, they looked like that. They were about 45 millimeters in carapace width. And here is a 24-year data set. 98 was an El Nino event. Not much is happening. What we're looking at is young green crabs recruiting to the estuaries. And a good year class in one estuary is a good year in all the others, and vice versa. So actually, there are two phases to this graph. The first phase and the second phase. In the first phase, recruitment is, hit and miss. There are certain years when you get recruitment. 205, 206, 2010 was a mini El Nino. And then we have 3 years of complete recruitment failure. Imagine what I felt like, setting traps and not getting any. I said, Sylvia, you need another project. You can't do this. You're crazy. But luckily I thought, well, I can demonstrate their local extinction, right? But then that never happened because 2016, 15 and 16, we had another El Nino. We had good recruitment. And after this event, we have good recruitment every year. So in this period, we did not have a huge breeding population in Oregon estuaries. So we were totally dependent on larvae coming to Oregon in the Davidson Current. But then after that, it looks like there are multiple larval sources. Now, here we are superimposing temperature on this graph. This is winter temperature off the Oregon coast, off Newport. And you can see warm winters, there's a chance for a good year class. 2005, 6, 2015, a mini El Nino, and then 15, 16 are El Nino years. And after that, we get good recruitment every year, even in 2020, when we shouldn't have gotten any larvae from the Davidson Current, right? So what's going on? Where are those larvae coming from? Well, looking at the same data, ocean surface temperature off Newport during the winter, and now we're looking at an average of abundance of green crabs in all the estuaries. And you see in the first phase, we get a real nice correlation between year class strength and winter water temperature. So 61% of the variability was explained by regression. So the warmer, the greater the chances of a good year class. But then we're adding the more recent years to this graph, and we find that 17, 18, 19, 20, 21, 22 are outliers, and our regression is not as good. So we're getting more young green crabs recruiting to Oregon estuaries than we would have predicted by our first hypothesis that the Davidson Current was the mechanism for recruitment. Now, here we are looking at the strength of the Davidson Current, and we're looking at southern copepod abundance. So when there's a lot of southern copepods in Oregon, that means the Davidson Current was very strong because it brought them to Oregon. So it's actually like a proxy for the strength of the Davidson Current. Again, phase 1, very good regression. The stronger the Davidson Current, the greater the recruitment. 69% of the variability explained by regression. That's pretty good. But then look what happens when we add the more recent years. That relationship is breaking down. So our first hypothesis that larvae arrive from California in the Davidson Current is not the only source of larvae anymore. Now we probably have additional larval sources after that second El Nino, the 2015-16 El Nino, from breeding populations in Oregon, Washington, and British Columbia. Here's another clue that the Davidson Current may not have been responsible for all the green crabs that we recovered. In phase 1, the size frequency distribution at the end of the first growing season is very similar to this, like this. Crabs range from 30 to about 55 millimeters with an average about 46. And now we're picking up a lot more smaller crabs. And these probably did not come in the Davidson Current because they're too small at the end of the first growing season. And they could have come from the north. Because when the Davidson Current dissipates at the end of the spring, we have the shelf break current that flows from British Columbia, hugs the shore along Washington and Oregon, and the larvae can arrive that way. And we have very lucky that we have some evidence for that. Carolyn Tepal, who has been studying green crab genetics for over 20 years, she's at Woods Hole Oceanographic Institute. She found that the green crabs on our coast are pretty well homogeneous, genetically homogeneous, except for one population on Vancouver Island, the Souk population. It's an inlet, it's quite isolated, so some green crabs got in there, and after a number of generations, they developed their own unique genetic fingerprint. And we've been sending Carolyn samples of our crabs for many years, each year, and she found that Macabe, Willapa, Tillamook, and Netards each had crabs with the Souk genotype. So those crabs had to arrive as larvae from the north. We also have evidence that we can get local reproduction. Alan Shanks at Oregon Institute of Marine Biology was sampling plankton in Coos Bay, and he found that in January, February, and March of 2010, which was a mini El Nino, he found first instar zoea larvae from green crabs. Now, those had to be produced locally. They could not have come from California. They were just way too small, too young. So now we have evidence for multiple larval sources for green crabs that come to Oregon. And we don't know what the relative contribution of these sources might be from year to year. But it does look like green crabs are here to stay. Thank you. I want to acknowledge some of the people that my collaborators in Oregon, Washington, British Columbia, and Woods Hole, and I'd be happy to take questions.
Thank you, Sylvia.
Yes.
How long does it, that was very interesting. How long does it take to go from zoea to reproductive adult? Is
Oh,
it
okay.
1 year?
From zoea, maybe a couple months at 12 degrees to be a small crab. And green crabs live for about 6 years. And to be they reproduce in their first year on the mudflat.
Yeah.
Yeah. Those little guys that I saw in that little Tupperware container, they were reproductive. Actually, they were in the sunshine and they were trying to mate.
Do you think that they're showing adaptation to the colder waters and that's why in 2020 it didn't knock them down as much?
I think the 2020 ones, I think what might happen is I might have gotten a little confused about the sizes. Maybe they came in we had recruitment from the north the previous year.
Mm-hmm.
Maybe there was, I have to check that because, you know, the ones that come from the north would be smaller, right? And I may not have picked them up in the fall. They might have been too small. And then they looked like a huge reproduction, but they came from a different source, maybe.
Okay.
I will never know. I mean, I can look at the size frequency distributions and look at maybe some records of water movements, you know, whether we got a lot of water coming from the north that year or not.
Yeah.
Yeah, it's frustrating because you can't really figure it out. Yeah.
Wonder, I can't say 2020 and not think of COVID. Was like the absence of human activity, could that explain any of the additional crabs?
Oh, and that's I don't think so. No. Because I was still going out there doing the same thing. Yeah. I don't think so.
Yeah, good.
I don't think that COVID had anything to do with it. No, I don't think so. It's a good hypothesis, but I don't think so.
Awesome. Well, thank you so much, Sylvia. And if anyone wants to see the crabs after the end of the talk, she brought them. Okay, so our last speaker is Stacy Bronson. Gonna wait for your title to appear. So she's gonna talk about principles of A&P by 25X and a lightning tour of our interactive lab. Yay! Thanks, Stacy.
I'm gonna go like Nate and go very quickly. So really quickly, the BI 25X series is a 3-term series of anatomy and physiology that I developed that's what I was hired to do and developed to now teach. It's a 5-credit lecture lab combo that is based almost entirely off of Lindsay's 200 series, the 2-3X series. The content is all from the existing courses, and I can't thank or compliment Devin and Lindsay enough for all of the work that they've done in developing these in-person series over the last several years. I relied heavily on them for what they talked about, how they talked about the different labs, the things that they've done. We really tried to make these two, Lindsay's course, the 2-3X and 2-4X, as similar to mine as possible. There are 25 labs in total over the course of the 3 terms, and we took them from in-person to remote, as we all made everything remote, and then I further adapted those to make them more permanent for eCampus. So a lot of the footwork was done by the teaching team, all the GTAs that were working for us within the labs, and the mad dash that we all took during that first year of the unmentionable. So I have a variety of different types of labs that we teach. Some of them are student-led explorations, some of them are observational, some of them are 3D modeling, and I have them work on collaborative activities. So I have a whole bunch of different types of labs. And if you're interested, I actually have a whole write-up of every single lab and what style it is. So one of the 3D models that I bring up is called the bone box. And this is really so that they can learn the skeleton, the human skeleton. And in the lab, this is what the bone box would look like. It is a big trunk full of disarticulated skeleton. It's just plastic bone, a model of bones. So that's what they see in the lab, and we give it to them in the lab, and they just take it and explore the bones. So it is not put together in any way. How the bones get put back in the box just depends on who put the bones back in the box. There's no sort of like rhyme or reason to how it all goes in there. And the students are to take the bones, we give them a list of terms that they need to know, and then they have to go to their resources to figure out how to actually identify those parts on the bones. So on my eCampus version, we scanned every single one of those bones using a 3D scanner. So these are actually the bones that are in the lab. And when they open up the bone box, they see a bunch of thumbnails, and they're all shuffled. Every single time they open it, they're in a different order. So they can go through and look at all the various types of bones. And when they find one that they want to look at, they can click on it, and that loads the model for them, where they can then zoom in, zoom out, rotate the model. From there, it's their job to look at the list of terms that they need to know, go to their resources, and find out what that is and what that is, and be able to identify those for the assessment later on. So that's one of the labs with exploring the human skeleton in 3 dimensions, which I'm planning to do a research project on through eCampus. So stay tuned for results of if it actually works. An observational experiment example is one where I perform the experiment in the lab. We can't ship this lab equipment home to them and have them wire themselves up. It's just not cost effective or reasonable. So some of our experiments we've recorded in the lab with myself or Lindsay or my husband as subjects for the lab, and then we provide the students with data. So this is one of them. It's an example experiment where I'm teaching them about lever arms and the distance from the fulcrum and how much muscle activity is required for the same load. So the load isn't changing. I'm scooting it closer and closer to the insertion of the deltoid, and they're watching the real-time data from the EMG activity as I move this from spot to spot. And then once they've seen this as they would see in the lab, only maybe a little bit less problems with the software. Then they go ahead and run the analysis for them using the cursors to pull out the data and tell them what the EMG activity is for each section. From there, they take this data, they create their own graphs, and they answer questions about it. So I can't send this lab equipment home to them, but they can still feel like they're a part of the experiment and look at the data. Another one is laboratory simulation. So instead of having them deal with mice, we can actually just do a media version of the simulation. So I have an endocrine lab where we look at the difference in metabolic rate in rats based on whether or not they have a thyroid or a pituitary gland, and what we inject them with, so we can measure their metabolic rate. I also have a number of digestion labs where they take an enzyme and a macronutrient, and they put it in and they watch and see if the enzyme works, if it breaks down that macronutrient into its components. So this is a quick overview of what they might see when they're looking at the rat lab. They select in any order that they want, what rat they want, and then they go through and they tweak the airflow, they measure the amount of oxygen consumed by the rat over the course of the 1 minute, and then they can palpate for a mass on the thyroid, and then their data populates into a table for them automatically. Once they go through all the way through the experiment, they can actually download that data and then look at it and analyze it and figure out what happened during that time. So just a little bit of a simulation for them in order to see what it would be like if you were to do an experiment with animals without actually poking them or touching them or sending them rats to deal with. In this particular one, I have 2 options for them to walk through this lab. They can either do the whole lab themselves and try to make sense of this data themselves, or they can do a video guided tour where either way, they see the lecture about what the metabolic pathway is and how the different hormones work on metabolism. But if they take the video guided tour, I've chunked that lecture up to talk about one piece. Then they run that segment of the lab, look at those data, and then I give them sort of the rundown on what that all meant. So they can choose their own adventure in a few different ways on this particular lab. The new labs that I just created, one of them is for blood typing. So this is something that we have in the lab currently that works very poorly. And this is a simulation that we've built up for them to go through and be able to actually do the sampling. So I run this in kind of super speed. Right now, they can't make any mistakes because it's the first version of this lab, but they go through and they add blood to the wells, they add the various anti-serums to those wells, they mix it up and wait for either coagulation to happen or not happen, which then helps them identify the blood types. So once they finish all of that, they get them all mixed up, time goes by, things change, and then they're able to make their guess at what the blood type actually is. I also have built out a quiz tool for them so that they can guess the blood type based on pictures, and they can continue to jump through and guess blood types, as well as have a reference table for what is in each of the blood types, and then answer questions about them that populate live with the answers so that they can practice these exact exam questions over and over and over again as much as they like. Blood typing happens to be one of the things that at least my eCampus students struggle with a lot. So this tool, they've already told me, changed their understanding of blood typing. And blood pressure is another one that I just finished creating. And in this one, you have a waiting room full of patients, and you can select any of the patients, and you head in to place the blood pressure cuff, place the stethoscope, and then actually take the blood pressure. I didn't have the audio playing for this, but there's background noise, there is wiggling of stethoscope noise, there is that noise, there's that noise as you deflate the cuff. There's blood pressure that starts off strong and gets quieter as it fades out for the end. And then if you're nice to your patient, once you've heard the bottom, release the valve quickly, input your pressure. And if they get it close enough, it gives them the correct, and then it shows them what they just recorded and what the actual blood pressure was. So close enough is close enough, even in cardiology, where I worked for years, taking blood pressures and running EKGs. So in this case, they have a pretty good parameter of error that they can make, but it always tells them what it actually was, so they can go in and try again. I have one more experiment to tell you about. It's the P Lab. You may or may not have seen students walking around quarterly with beakers, seeming a little bit shy. During the pandemic, the TAs worked up a way to do this as a group crowdsourced experiment at home using pint glasses and measuring cups to create their own graduated cylinders at home or their own beakers at home. And then we have pictures that they can actually compare their urine color. So we want them to look at urine concentration over time, depending on whether or not they drank anything. And they self-assign to various treatment groups. And if they don't want to do the P Lab at all, they can be a data curator. They measure their urine output for every 2 hours or every 30 minutes for 2 hours, and then they record their data and interpret the results. So this is what the Google Sheet looks like. And this is how that they can tell their urine concentration instead of using the actual tool that we use in the lab. And then their data powers the graphs, and they can see that changing over time. And I'm running this as a longitudinal experiment because I've got my 2021, my 2022, and now my 2023 data all in the same spreadsheet. So it's growing, the sample size is growing, and the graphs are smoothing out more and more how I think it should actually look. So it's fun for them to be a part of that experiment and provide this. All of these are open source. I've created almost every bit of material for my anatomy course as open source material. So I told them from the get-go, open source, this is what I want. I want all of my images to be open source, all of my labs to be open source, all of these tools to be available. So along with the open resource textbook that we use, all of the labs are accessible, and been sharing those out with a number of different colleagues at different universities. So that's a little bit peek behind the curtain of interactive labs in anatomy.
Awesome work, Stacy. I really like the simulation and simulations and the thing that it is open source, that we might be able to use that in our other courses like introductory biology campus courses as well. So my question for you is, what kind of assistance do you have from the eCampus? How long it takes to make a simulation like this?
Sure.
What's your role and things like that?
So there are various lengths of time. The blood pressure and blood typing one that we just did, we worked on it for about 6 months. And it was every other week meeting kind of back and forth between them. The bone box took 1.5 years. The endocrine lab took maybe 4 or 5 months. And these took about 6 months because we were working on both of them simultaneously. When we actually got into the blood typing, I think it only took us 4 weeks. Like it was pretty fast because we had been working on blood pressure for so long and they used a similar platform. So it kind of depends. And then in terms of your question of how much support do I have from them, anytime I ask them to make a new lab for me, they send a new MOU. They're like, yeah, here's more money. Let's make more labs. And me and the media team get along very, very well. So it's been very easy for me, which I hear has not been the experience for all other people. So as long as there is potential to continue improving the course and they have the resources available to work on it, it sounds like I can keep just asking for more labs and they'll keep making more labs for me. And I really like to push when like the blood pressure lab, I pushed pretty hard about because I was like, well, the 20X or 22X, they mail blood pressure cuffs. And if we can create a good enough version of this online, they don't have to do that anymore. And you automatically drop $50 or so off of every student's lab fee. So they were like, oh, well, yeah, let's do that. Yeah.
So I was curious from like the students' perspective, like they're logging into campus, they see this, do these different labs take them to like another website, or is this all sort of embedded right within the campus?
So I have it both ways. I have the labs embedded within the Canvas shell so they can interact with it right there, which is a little bit smaller of a screen. And then below every single one of them, I have the external link so that they can pop out into another browser and work on it that way. So I have both options empty. They're all version 1 at the moment. I have changes for every single one of them. So at some point, when they're ready to give me 1.1 or in some cases 2.0, we'll revisit them because there's certain things that are missing, I think, still, like in the bone box, I really want to scale on the bones. And there's 200 and something of them. So it will take a while and they need to allocate the resources to have them go into every single one and add scale. But yeah, little tweaks and things happening along the way, but
Those are amazing. Thank you so much for sharing them.
Curious, like the bone box took so long. Would doing it again be quicker? Like I can imagine scaling that to say you want to do, let's say, a different organism.
oh yeah,
Or like
it would be much faster now because the bone box, they actually had to develop the entire web-based app and work with Sketchfab in order to host things without labels and names and logos and things on them. So it's now built and behind the scenes on the bone box, they also built me flashcards and an annotation configurator so I can make quizzes out of them. They built me a whole bunch of stuff on the back end to use as an instruction.